major that these two instruments is first installed in this country, and thereby the health sector of Bangladesh, I think the facility will appear that is helpful for next the medical profession who are trying to do research in this lab. The objective too is that capacity building. As you know, the genetic field, human disease-related genetic field, that is new in this medical members. So we are trying to develop our faculties and professional staff. And here you see some of the exposure, foreign exposure training we got from well-renowned universities, and on inside training, that is side training, and international exposure visit, that you see that these are the sons of this land who are actually work in different renowned universities, that is Harvard University, Lausanne University, Switzerland, Dublin University, and National University of Sport. They visited here several times. We worked that one of my friends who are working at Switzerland, he actually come several times here to facilitate our manpower development. And some of the workshops also going on for faculty development and finished. Actually, these are on several aspects. One thing is that some of the invasive method of molecular diagnosis and some of the workshops for diagnosis of genetic disorders and their management are actually was done through this project. And objective 3 was establishment of the genetic counseling center. You see some of the focused disease we addresses, and actually we developed some genetic counseling structured format. We actually developed a very important that advanced genetic curriculum that is published in the International Journal of Multidisciplinary Research and Review. Advanced genetic curriculum for Bangabandhu Sheikh Mujib Medical University. These are that actually I have mentioned earlier that this manuscript is submitted, and we got, in addition to 52 genes, as two of the genes that breast cancer associated gene 2 and zygote arrested one live gene, we get 4 new mutations in our population. Some of the research are going on. One is epidemiological data of breast cancer patients, and another vital one is microRNA of profiling of Bangladeshi humans with breast cancer. And I have already mentioned that human cardiomyopathy related genes, multi-genes analysis are going on, and these dreams are now comes to true with the fund of HEQEP. And sustainability, it is very crucial that Bangabandhu Sheikh Mujib Medical University is sufficient enough to provide service in these regards, but for the further development of these technologies, we actually think to collaborate with the pharmaceuticals or international experts. And in this regards, for continuation of the research work, I mentioned that to develop a center of excellence at Bangabandhu Sheikh Mujib Medical University at this South Asia region, we require further research grants. Thank you for your patience there.
Dear participants, as you can see, I am running a little bit late. We are supposed to go on a break at 1:15, but I will take only half an hour because I have three more presenters. So if I do the math, it will take me 27 minutes. So I hope you will bear with me these 27 minutes. Our next speaker is Professor Dr. Muhammad Tufazzal Hussain. Tufazzal Hussain is also from Bangabandhu Medical University. Then it is incorrectly written, Bangabandhu Agricultural University. And Dr. Hussain will speak on enlistment of facilities to promote quality higher education and research in biotechnology.
Our target, we developed 3 modern laboratories, microscopy and genetic engineering, molecular biology, cell and tissue culture, approximately 2,200 square feet. These are some photographs you can see. And this is the very fancy lab, and you can compare globally standard laboratory. We, from our own experience visiting across the globe, the biotechnology laboratory was designed. And we purchased modern equipment, including a real-time PCR, which is very expensive and was a very big dream for us. And our students, you can see the photographs, they are using the laboratories and doing the productive research. And I welcome the other members who are working in biotechnology to utilize our facilities, especially the real-time PCR. And we procured the textbook, the top-class world textbooks for our students. And we developed 25 protocols in our laboratory. Some of them are optimized to conduct the modern research. We established a bank of 600 beneficial bacteria that we isolated from our environment. And nearly 15% of the bacteria we found that they are new species. So this is a very big, ambitious project under we call Mining Bangladesh Bio Gold, and it is going on. And 65 isolates we identified through molecular techniques, including 16S rRNA gene sequencing, and 28 isolates we tested in the laboratory greenhouse, and some of them in the field conditions showed that by using those big bacteria, we can save NPK fertilizer about 50%, which would be a very big contribution in the field of agriculture. And we, some of our isolates showed tremendous activity in suppressing plant disease that could be utilized to develop biopesticide. And we have been studying the mechanism of their function, and we sequenced 4 important bacteria. Whole sequencing and gene annotation has been done, and we are now doing the post-genomic work to develop, to understand the molecular mechanism and to develop powerful strains for commercial use. And we published 22 papers in international journals and 11 book chapters. We edited a book, and 21 students completed their MS thesis in our laboratory. And this is our the website. It is a complete website, a dynamic one, and we introduced e-learning facilities by using Moodle software. So all the courses are now online, and students can submit their assignment and do everything, interact everything from their home. And this is the our website is very interactive, and you can go there. These are the other faculties and students are trained by a series of workshops we organize in our university, as well as some of our faculty members they attended in international conferences in abroad. These are some photographs. These are also, we organized an international colloquium in response of our visit to University in Nottingham, Malaysia. They came to our university, and we jointly organized an international colloquium on curriculum and research in biotechnology. Okay, and these are we have developed good facilities. This is why we got many collaborators across the globe. This is a USDA-funded project collaborating with University West Virginia in USA. Okay, these are our collaborators across the globe. We have the collaborators, and we are doing effective research. Okay. And one of the interesting findings in our research is discovery of some probiotic, plant probiotic bacteria, as I mentioned, which we discovered from the indigenous rice varieties, can reduce the fertilizer cost of about 50% is rice production. And by using half dose of fertilizer, we found that bacteria can save the 50% fertilizer and give the higher yield than the full doses of fertilizer. And we are now talking with some industry to commercialize this technology, to scale up the technology to produce biofertilizer for our farmers. We found the same result in wheat by bacteria isolated from the wheat. We got, strawberry is newly introduced crop by Dr. Manjur Hussain, he presented earlier. The most dangerous issue is that in strawberry, farmers do not know which pesticide they should use. They use a dangerous pesticide, a cocktail of pesticide. This is why we developed some bacteria which can, replace the synthetic pesticide and produce safe strawberry for us. Yes, sir. These are the biopesticides that we have trained. Our colleague, he developed the PCR detection method to identify field diseases. These are the journals where we publish the papers, which are all very high impact journals in the world. These are some journals, okay, I'm going fast. We discovered 21 new antibiotics from our antagonistic bacteria. And whole genome sequencing, I mentioned 4 bacteria we did, and we annotated, and now gene expression also we are studying by using our real-time PCR, especially the functional genes involved there, so that we can produce a transgenic bacteria which can compete with the synthetic chemicals, replace the synthetic chemicals in the agricultural field. And we developed this protocol. How can we, commercialize our technology with the industry partners? Which part industry partners do and which part we do? And we received I received the Bangladesh Academy of Science gold medal in the biological science senior group last year, and also UGC Research Award. And we are trying to make a collaboration with industry and farmers. Here you can see a picture to use our technology in the farmers field. And our vision is very, now I am at the end. Our vision in our sub-project, it was mentioned that if we can develop such a facilities, state-of-the-art lab facilities for biotechnological research with trained manpower, then we can upgrade our department into Institute of Biotechnology and Genetic Engineering. And our Vice Chancellor here, he is leading to set up such an institute, IBT, like IBA at Dhaka University. And I believe that institute will be a center of excellence in biotechnological research, especially in the field of agriculture. Our developed biotechnological products, especially biofertilizer and biopesticide, we believe that if we can properly commercialize, then it could be, give us a second green revolution, reducing the, agriculture cost. In agriculture, the most striking problem is now input cost, especially in rice. For production of 1 kilogram of rice, our farmers use 25 taka or 26. This is why agriculture is not profitable. We use incredibly high amount of fertilizer, and most of the fertilizers we are getting from the abroad. This is and some urea we can produce only by burning the methane gas. So if we can introduce biofertilizer and biopesticide, our products would be the safe, and our environment would be safe, and production would be decreased, and it would be sustainable. I am thankful to the HEQEP, because HEQEP help us to realize our dream, and I hope the UGC will help us to patent our technology. Thank you very much.
We'll do our best. Professor Tafadzal Hussain, thank you very much, and I'm sorry, you are from Bangabandhu Sheikh Mujibur Rahman Agriculture University, Gazipur. I'm sorry for the mistake. Thank you. Our next speaker is Dr. M. Kobiruddin. Professor Kobiruddin is from Jahangirnagar University. He will speak on, his CP number is 2334. He will speak on strengthening postgraduate research by establishing wastewater research unit. Dr. Kobiruddin, please.
The cause also we know, the high cost of effluent plant, high operational cost, unwilling to take extra expenditure, dependence on the foreign export and technology, bad actions of the Department of ETP, and lack of monitoring from the authority, and inside the other thing, because not properly monitored by the authority, especially the government personally. How the situation will be changed? Develop indigenous capacity, awareness, extending the monitoring system, proper training, the persons who operate the HEQEP, proper selection of technique, think for longer period, not for short time. What we are doing actually, as I mentioned, since 2000, we are trying to do something on wastewater treatment. So model plan, as I showed you, we developed many absorbing materials, which is very suitable to treat the water. What the outcome from this project? Established of Water Research Center with advanced research facilities. From this money, we actually by TOC, HPLC, UV visible spectrometer, fluorescence spectrophotometer, milling water plant, and other relevant instruments. Development of wastewater treatment plant, support 4 postgraduate researchers, conduct training and workshop, assist the adjustment in industry in remedying pollution. This is the plan to be developed. What is the practice actually? Lack of technical knowledge, high operation cost, high chemical consumption, high manpower involved, technical fault of ETP to pour after sales service from the suppliers, dependence on the foreign expert in technology. Objective of the Water Research Center: extending our product with our graduates, collaborate research network between research institute, public and private enterprises, develop pilot-scale ETP facilities to introduce new techniques for wastewater treatment and process optimization, provide training facilities to the personnel involved in the wastewater treatment plant, established well-equipped laboratory. So this is the laboratory which we actually established, and this is part of other instrument. This is our instrument. So we actually conduct 3 workshops. This is one of the pictures of this workshop. So this is pilot ETP design. Especially this, as I mentioned, comprehensive treatment plan. And this treatment plan, we set every option, physical, biological, and chemical. As I mentioned in the video, actually, in generally, the industry is set a ETP with all options, physical, biological, chemical. But not all options are needed to treat the wastewater. So we can give information to the industrialist which part of the actually option is needed. So we design with bulb system, and we can use any of the options, physical, chemical, biological, or any of the part we can use, and which is needed for that industrial wastewater, they can get information. These are the publications published from this project. Service and training. Industrial wastewater, now we can actually provide support. Now actually we are supporting our nearby industries. Regular monitoring, and also if they face any troubleshoot, also we are capable to solve their problem. So, as I mentioned, environmental aspects, we have to now we are capable to actually sustainable environmental system developed by this laboratory, and we can support training and operation management system to the personnel. So this is not actually related to this project, but we developed this waste management system in Jahangir University in 2012. So just I will conclude by this waste management. Okay, just I will finish it. So this is a problem we can solve. Why I showed this thing actually? Because now our group can capable to solve 2 types of waste management: solid waste and also liquid waste. So I showed that plan. So upcoming challenge. As I mentioned, we are now sufficient in research, and also we have two plans. One is related to liquid, that is wastewater, and another is related to household waste. So now this is practicing in Jangir University. We are providing from that solid waste management 30 household gas, and now we are supporting the wastewater treatment to the nearby industries.
Thank you.
Almost 3
Thank
years.
you.
So,
Thank you.
So this is.
Thank you,
for
Dr.
your attention.
Kobrudin. Thank you for the presentation. Dear participants and distinguished audience, we are left with just one presentation. Is Professor Muhammad Yunus. Professor Muhammad Yunus is from Shahjalan University of Science and Technology.
To achieve our goal is to do research on materials chemistry. We set goal objectives to do research, renovation of the existing research laboratories, procurement of the state of our instruments, exposure visits, conference and training, finally to do the research on materials chemistry. So in the next two slides, I'm going to show you our innovative laboratories. You can see we actually installed remotes and the fire extinguishers, electrical connections, steel finishes, all other things related to the laboratory. And then I'll show you some state-of-the-art instrument. This is the fluoride optical microscope, and you can see the optical data that we are to identify the differentiation. The next one is thermogravity analyzer. By using this, we can actually determine the thermal stability of polymer and materials. The next one is gel formation chromatography system. We use this instrument to determine the molecular weight of the materials and polymers. And now I'm going to show you some small equipment, such as infrared spectrometer, rotary evaporator, then gas chemical for doing reaction on the nitrogen, and you can see secure shell to store solvents, chemical solvents in the cabinets. Now, so this is the next part of my talk. You see, we organized a conference on materials chemistry, and the theme of the conference was materials for innovation. And Professor Christopher Mola, the Department of Chemistry, gave the theme space. We have more than 100 participants, and here is some activities of the conference. We also organized a discussion meeting on the progress of prospects of science education in Bangladesh. Now I'm going to show you some exposure visits. We actually visited Indian Institute of Technology, Guwahati, Assam University, Center for Advanced Research of Sciences of Dhaka University, and CSI of Bangladesh. We actually established collaboration with Assam University and IIT Guwahati through this exposure visit and conference. So you can see the workshop we organized, all the instrument we purchased are state-of-the-art instrument. So we need to know how to use them, how to manage them. So we actually organize workshops and training programs. And you can see we organize workshops on liquid crystal identification, differential scaling capability, spectroscopic methods, and gel formation chromatography system. And also, we invited expert professors in our field to actually know the advances of material sciences. And now, the next few slides, I'm going to show you the research that we have done under this project. So you can see some molecules and some polymers. On the left-hand side, they are molecules. And you can see they could be either superplanar or octahedral, depending on the manner. And you can see they are conjugated core on it. And from monomer to polymer, you will then goes longer wavelength. And then I'm showing you some precursor molecule. They are not commercially available, so we have to do these reactions to get the precursor molecules, and we have to do all these reactions under nitrogen. And the left hand side they are acetylene ingredient, and in the right hand side they are partner precursors. And there are some research activities, and you can see if we use monocarbon acetylene, we get monomers, and if we use dicarbon acetylene, we got polymers. And you can see you can determine the average molecular weight of the polymer. And here is the crystal structure of a monomer, and you can say that from this monomer that the polymer derived from this the one-dimensional and physical. And we determine the properties of this polyion by absorption and polymorphism. I will discuss in the first one. You can see the dotted line is the absorption spectrum, and the dashed and solid line is the photosynthesis spectrum. The dashed line is the fluorescence, and the solid line spectrum is phosphorescence. So due to the presence of platinum, we get phosphorescence of this polyion. Now, why actually we use platinum? So if we use platinum, we have metazoic and transfer. How many minutes do I have? Okay, fine. So we actually do nonlinear optical determinants. The physics laboratory of Shahadam University. Professor Yasminov project also funded by HEQEP. So this is the nonlinear responses of one of our compounds. So from monomers to dimers, we see higher responses in the nonlinear activity. And the next slide, I'm going to show you once again why platinum is important. So if we use platinum, we have single emission and triplet emission, and we have 4 times electroluminescence efficiency. That is important for LED characteristics. You can see that the LED device is here, and polymer is sandwiched between anode and cathodes. So this is a new research in our lab. Once again, this is for polymer synthesis, and this is the polymer molecular weight of the polymer done by GPC, which is probably from this research project, and this is the introduction of platinum in the polyion. And now, next I'm going to show you how we are going to introduce liquid crystal in the polyion. So, crystal liquid, one is has got position and orientation order, and that has got no position and orientation order. But in liquid crystal, you can see there are no position order, but some sort of orientation order. Why actually we are going to introduce liquid crystal into LED? Because they actually give greater charge carrier mobility, and also they will give polarized emission. This is both as important in light emitting diodes. Now, I'll show you once again some properties of this material and NMR spectrum to show that we have made this compound. And finally, I'm going to show you the optical biretube this optical crystal by polarized optical microscope. We actually characterize the liquid crystal in combination with differential optical calorimeter and polarized optical microscope. And from differential optical calorimeter, we see the phase transition, and from optical detector, we actually determine the phase stability of this material. Finally, I'd like to show you, so this is actually the idea. We have made some of this material. Now we are going to optimize this material by taking the liquid crystal course. So finally, the achievement of this project. So new state-of-the-art instrument have been installed and commissioned in the renovated laboratory. We actually conferred some research degrees, and we established research collaboration with other universities. So we published a paper in international journals. Here are some list of journals that are in preparation, and also we established collaboration. You can see with Australia, India, and Bangladesh. And finally, in conclusion, I would say that further fund is required to sustain this research in our laboratory. So we need to have PhD at Kuala. Finally, I would like to acknowledge my students who worked hard to make the
Thank you, Dr. Yunus. We have seen that you don't need to read the names. Thank you very much. Thank you. Thank you. So that brings to an end to this part of the presentation. Thank you, distinguished guests and audience and honorable Vice Chancellors.
Ladies and gentlemen, Professor Dr. Muhammad Yusuf Ali Mullah, member of UGC, will chair in this session. And please have your seat, because we are going to start the second part of the technical session now.
Assalamu alaikum, distinguished audience. I welcome you once again for the second part of the first session. In the second part of the session, we have invited 7 vice chancellors to share with us the impact of higher education quality enhancement project in their respective universities. It would have been appropriate if we could invite all the vice chancellors to speak before you, but purely on time constraint, we could not do that. Anyway, I would now request the Vice Chancellors of Dhaka University, I'm not sure whether he is present, Professor AMS Arifin Siddiq, Vice Chancellor of Bangladesh University of Engineering and Technology, Professor Dr. Khaleda Ikram, Vice Chancellor of Chittagong University, Professor Dr. Iftikhar